Friends, myself Jagadish Kumar Karal, lecturer in zoology, KB DAV College, Niragarpur. Today I am going to discuss about genetic engineering or recombinant DNA technology. Friends, genetic engineering is the direct manipulation of DNA to alter an organism's characteristics in a particular way. That means we can alter or change the characteristic of an organism by manipulating or changing its DNA. Traditionally, humans have manipulated genomes indirectly by controlling breeding and selecting offsprings with desired traits. Genetic engineering involves the direct manipulation of one or more genes. Most often, a gene from another species is added to an organism's genome to produce desired phenotype or desired products. Let us go to the definition of genetic engineering. Genetic engineering is a technique for artificially and deliberately modifying DNA to suit human needs. That means, as per our needs, we can deliberately modify the DNA of an organism or change the DNA of an organism to produce the products as per our requirements. Genetic engineering is the process of inserting new genetic material or gene DNA into existing cells in order to modify its characteristics. That means, by the process of genetic engineering, we can insert new genetic material or genes or DNA into the existing genome of the cells or organisms, and the organism produce desired characters. Genetic engineering is the process of altering the genetic structure of an organism either by removing or introducing DNA. That means, we can add the DNA into an existing genome of the organism, or we can remove the genes of the genome of an organism. Genetic engineering is also known as genetic modification, genetic manipulation, molecular cloning, and gene cloning. Let us go to the aim of genetic engineering. The aim of genetic engineering is to alter hereditary apparatus of a cell or organism to develop new strains. That means, by changing the hereditary apparatus or gene of the cell or organism, we can develop new strain or new type of cells or organisms. Another aim of genetic engineering to produce specific products or phenotype in the cell or organism. These specific products such as hormone, antibiotics, and vaccines are produced through the process of genetic engineering. Let us go to the history. Since 1917, direct manipulation of DNA of organism seems possible through genetic engineering. The term genetic engineering was introduced by Jack Williamson in his science fiction novel Dragon's Island in 1951. The first recombinant DNA molecule was made by Paul Berg in 1972 by combining DNA from the monkey's virus SV40 with lambda virus. Paul Berg is referred to as father of genetic engineering. He received the Nobel Prize in 1980. Recombinant DNA technology. Genetic engineering deals with alteration of the genetic makeup of cells by deliberate and artificial means. It involves transfer or replacement of genes so that new combination of genes or recombinant DNA is produced. So, this process is also known as recombinant DNA technology or gene splicing. Recombinant DNA technology is the joining of DNA molecule from two different species. The recombined DNA molecule is then inserted into a host organism to produce new genetic combination that are of value to science, medicine, agriculture and industry. Let us go to the tools of genetic engineering. First tool is the enzymes. Two enzymes used in genetic engineering. These are restriction endonuclease and ligase. Restriction endonuclease, otherwise known as molecular scissor, it is used to cut the plasmid and the foreign DNA molecule at specific points. That means it is used to cut the DNA molecules at specific points. The ligase is used to seal the gap or to join bits of DNA. That means ligase is used to join the fragment of DNA to produce recombinant DNA. Next tool is vectors. A DNA segment used as a carrier for transferring selected DNA into living cells, as for example plasmid. What is that plasmid is? Plasmid is extra chromosomal circular DNA present in bacteria. This is used as a carrier or vector in gene transfer. Polymerase chain reaction. It is a powerful tool used in genetic engineering. It is used rapidly to make millions of copies of DNA from a DNA sample. That means in order to produce more copies of DNA, we use polymerase chain reaction to produce large number of copies of DNA. Next steps in genetic engineering. The first step is identification and isolation of genetic material. In this process, cell containing the gene is opened and the DNA is purified. That means the cell is broken and the DNA is opened, it is purified. Later, it is screened to find out suitable genes. That means suitable gene is now separated from the DNA strand. Cutting of DNA at specific location using restriction enzymes. In this process, the genes are separated by using restriction enzymes to cut the DNA into fragments. We have already discussed that restriction enzymes are the enzymes which is used to cut the DNA at specific points. Next step is amplification of gene by PCR. We have already gone through the process of PCR. PCR is used to amplify the gene segment in order to produce more DNA copies. That means we take sample DNA which is not sufficient to carry out the experiment. So, in order to produce more copies of DNA, we use polymerase chain reaction to produce millions copies of DNA. Later, these segments of DNA are separated through gel electrophoresis. This gel electrophoresis is a method by which electric current is used to separate the DNA segments which are taken into a gel. Next step is recombinant DNA preparation. Isolated gene is attached to the plasmid by ligase to produce recombinant DNA. That means the gene which we have isolated, now we want to combine with a vector, that is a plasmid, with ligase, so that a recombinant DNA is produced. Next step is introduction of gene of interest into host cell or organism. The recombinant DNA is inserted into the bacteria because we have already produced the recombinant DNA by taking the gene we have selected. Some bacteria can naturally take up foreign DNA. It is the inherent character of some bacteria, they can take up the foreign DNA. Otherwise, bacteria can be induced via stress, either thermal or electrical shock, which increases the cell membrane permeability to DNA, so that DNA can enter into the cell nucleus. In animal cells, recombinant DNA is introduced using microinjection directly into the nucleus or through the use of viral vectors. That means microinjection is the technique which is used in case of animal cells to introduce foreign DNA into the genome DNA. In plant cell, DNA is inserted using Agrobacterium mediated recombination. The new genetic material can be inserted randomly within the host genome or to a target location. That means we can insert the genetic material randomly, or we can insert to a point which we have decided. Next step is cloning of DNA. The gene is cloned and the desired product is obtained. That means we have introduced the new gene and produced the recombinant DNA. Now, this recombinant DNA is produced multiple copies and produce recombinant proteins, which is obtained and utilized. As only a single cell is transferred with genetic material, the organism must be regenerated from single cell. The plasmid is replicated when the bacteria divide, ensuring unlimited copies of gene are available. That means we have already introduced the recombinant DNA into the plasmid by removing its plasmid of the host. Now, the bacteria is allowed to reproduce and a large number of copies of bacteria is produced. In plants, cells are grown through tissue culture. In case of plants, we can produce multiple copies of cells by tissue culture method. In animal cells, DNA must present in embryonic stem cells. Next process is downstream processing. As in the previous step, we came to know that the recombinant DNA is able to produce specific protein or desired proteins or desired product as per our needs. Now, it is the time to separate the product and purify it before it is used in the treatment or in other human use. These products are protein, antibiotics, enzymes and vaccines, etc. Now, here is a picture which indicates the process. You see, here the plasmid is marked. It is present in the bacteria. See, here is the bacterial chromosome. It is so that plasmid is extracellular chromosome. The plasmid is cut by restriction enzymes. Here the cut is marked by scissor. Scissor is not at all used by the enzyme, but the enzyme is used to cut at a specific point of the DNA. Similarly, in case of a human insulin gene, it is isolated, which is marked in red color. Then these two are combined to produce a recombinant DNA. Now, the recombinant DNA is introduced into the bacterium. In this process, initially the bacterial plasmid is removed and this recombinant plasmid is introduced into the bacteria. The bacteria accept it naturally or by means of some stress stimulation, either heat or electrical shock. Now the bacteria is cultured or it is allowed to reproduce. Large number of copies are produced. And this process is carried out in fermenter. Now you see, the bacteria reproduce to produce number of copies, and the bacteria able to synthesize the recombined protein which is deposited in the medium, right. These dots mark the recombinant proteins. Now the next step is followed. This is the fermenter, right, in which the bacteria is cultured. Number of bacteria is increased and the product is found in the culture medium. Then the product is separated and purified, which is known as downstream processing. Now they are bottled to be sold in the market for human use. Now what is the application of genetic engineering? Genetic engineering has application in medicine, research, industry and agriculture, and can be used on a wide range of plants, animals and microorganisms. Nowadays, genetic engineering has wide application in the preparation of antibiotics, vaccines, hormones, which are used in treatment of several human diseases. Genetic engineering potentially allowed totally new function to added to the capabilities of organism. It has opened up prospect for genetic engineering of industrial microorganisms, agricultural plants and animals to produce more valuable products. Thus, genetic engineering is undoubtedly the most significantly new technology in modern biotechnology. It is now widely used to produce valuable products to be used in the treatment of several human diseases, controlling pollution, treating wastewater, and produce several industrial products which are used for the comfort and survival of human beings. Thank you.